You think you know what lives in the ocean? Fish, sharks, whales, things you can see. But there's a parallel universe down there that plays by different rules. An entire kingdom that figured out how to vanish, not hide, vanish. Their skin, their bones, their guts, all invisible. You're staring right at them and your brain refuses to believe they're real. Glass sculptures drifting through pitch black. Alien pods pushing baby carriages made from corpses. Eels that look like ghosts of themselves. Fish with clear blood and see-through brains. Colonies that stretch 160 feet but stay invisible as dental floss. And they're not rare oddities hiding in some forgotten trench. They're everywhere. Millions of them. Some showed up on camera for the first time in 2025. Others have been eating our commercial fish stocks for decades and we barely noticed. Why does an animal throw away one of evolution's oldest tools, being seen? And what happens when the strategy that kept them invisible for millions of years suddenly stops working? Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from the Schmidt Ocean Institute's first confirmed colossal squid footage published May 14th, 2025, research from the Monterey Bay Aquarium Research Institute on barrel-eye fish mechanics published in Copia Journal 2008 by Bruce Robison and Kim Reisenbichler, and findings from the University of Oregon's SALP locomotion study in Science Advances May 15th, 2024. Now back to the information. 1,968 feet. The camera catches movement in absolute darkness, 30 centimeters long, about 1 foot. Transparent body rippling through black water. Hooks visible at the tentacle ends, sharp, curved inward. The crew realizes what they're seeing. Mesonicotuthis hamiltoni, the colossal squid. First confirmed live footage in human history. Not a photo, not a dead specimen. Swimming, alive, functioning. 2025 marks exactly 100 years since scientists first identified this species back in 1925. Maximum adult size estimated at 7 meters, 23 feet. But this is a juvenile, still growing. Marine biologist Kat Bolstad from Auckland University of Technology in New Zealand examines the footage, confirms species identification, the hooks are diagnostic, the body structure matches. This animal lives exclusively in Antarctic and sub-Antarctic regions. Water temperatures between minus 1.8 and plus 2 degrees Celsius, different from the giant squid Architeuthis ducks that roams other oceans worldwide. The colossal has hooks along the middle of its 8 arms. Giant squid lacks these mid-arm hooks. The colossal lives deeper, colder, darker. Sebastian captured the animal at exactly 600 meters. Temperature near freezing, pressure 60 atmospheres. The body ripples like liquid mercury. You can see the internal organs through the skin. Digestive system, muscle structure. The transparency isn't complete. There's structure, shadows, but light passes through most of the animal. An organization called Colossal captured similar footage during 2320, 24 Antarctic campaign, but low resolution. Features not discernible. Can't confirm which glass squid species. This Falkor footage? High definition, every detail visible. 2025, giving us creatures we've only known from dead specimens. Hauled up in fishing nets, torn apart, chewed up in sperm whale bellies, whale stomach contents analyzed, squid beaks counted, never swimming free in habitat until now. The glacial glass squid rising from broken ice. January 20th, 2025. Same vessel Falkor. Different expedition weeks earlier. A massive iceberg breaks away from the George VI ice shelf in Antarctica. The calving event designated A84 by researchers. Size roughly equivalent to Chicago. The Bellingshausen Sea suddenly reveals a stretch of seafloor hidden for potentially decades under ice. New ecosystem exposed. The Falkor deploys Sebastian remotely operated vehicle again into unexplored territory, 687 meters down, 2,254 feet. Temperature hovers near freezing, 1 degree Celsius, maybe less. The ROV lights sweep across water column, hit something that barely registers on camera, almost invisible even with artificial lights. Galateuthis glacialis, the glacial glass squid. First confirmed live observation of this species ever. This species was originally described back in 1906. 119 years of knowing it existed only through dead corpses pulled from nets, specimens that fishermen found, bodies that washed ashore, remains discovered in whale stomachs. Dr. Tom Linley from Museum of New Zealand Te Papa Tongarewa sits in ship's control room. Watches screens as creature performs what researchers immediately call a cockatoo pose, arms spread upward above head like a parrot's crest, defensive posture or hunting position unclear. The entire body completely see-through except for sharp hooks located at the tips of 2 longer specialized tentacles. These tentacles extend, retract, search for prey. The middle 8 arms completely lack hooks. That's the diagnostic difference from colossal squid. Taxonomic identification certain. These squids typically live between 700 and 1,000 meters depth, but warming ocean waters, documented by multiple research teams, push them into shallower zones. Thermal tolerance tested. Habitat range shifting. Ocean census researchers aboard Falkor record the animal using gentle rhythmic pulses to float upward. Energy conservation critical in water that runs consistently 4 degrees Celsius or colder year-round. Movement resembles underwater ballet according to expedition notes. Soft, silent, nearly invisible even under direct ROV illumination. The transparency so complete that finding these animals before remotely operated vehicles developed was almost impossible. Standard fishing nets? They slip through. Their jelly bodies offer minimal resistance. Don't show on most sonar frequencies. The ice breaking from climate change opened a rare window. For a few hours, humans got to see what's been drifting there all along. The ecosystem beneath revealed species never documented in this location, food web connections unmapped. Also, comment from which city you're hearing this from. Monterey Bay Aquarium Research Institute researchers Bruce Robison and Kim Reisenbichler published their findings in Capella. The eyes rotate inside a transparent shield filled with fluid. The shield covers the entire top of the head. Most illustrations and museum specimens never show this because the structure destroys when nets pull the fish up from depth. And Barry's remotely operated vehicle Tiburon films the animal at depths between 600 and 800 meters off Central California. 20,000 to 2,600 feet down, the fish hangs motionless, horizontal body, eyes aimed up. The tubular eyes glow bright green under ROV lights. The green pigment caps each eye lens. Researchers hypothesize it filters surface sunlight and helps detect bioluminescent prey directly overhead. The fish spots a drifting jelly. The eyes rotate forward. The body pivots vertical. Feeding mode. The transparent dome stays intact throughout, protects the eyes from stinging tentacles while the fish picks organisms off siphonophores from the genus Apolemia. These colonial jellies grow over 10 meters long, 33 feet. They trail thousands of stinging cells. The barrel navigates through them. The rotating eyes track prey. The shield blocks nematocysts, 15 centimeters long average, large flat fins for stability and precise maneuvering. The dome gives the animal a submarine appearance. The actual eyes sit behind the face. 2 small spots above the mouth are olfactory organs. Nares, not eyes. The real eyes are the glowing green tubes inside the head, first photographed alive in 2004. Specimens caught in nets survive hours in shipboard aquariums where researchers confirm the eye rotation. The fish turns from horizontal to vertical, and the eyes move with purpose, forward for feeding, up for scanning. What makes this jellyfish glow green when disturbed? He isolates samples in the lab. Low glow. He dumps them in the sink. Sudden bright blue flash. Not the sink. The calcium in seawater. But crystal jellies glow green. Not blue? Shimomura hypothesizes another compound. He names it green fluorescent protein, GFP. The jellyfish uses a photoprotein called aquarin that releases calcium and produces blue light. The GFP absorbs that blue and emits green. This energy transfer is called Förster resonance energy transfer, FRET. The jellyfish diameter reaches 5 to 10 centimeters in adults, almost entirely transparent and colorless. Up to 100 radial canals extend from a central mouth to the bell margin, up to 150 tentacles in full-grown specimens. The tentacles have nematocysts for prey capture, no effect on humans. Between 1961 and 1988, Shimomura collects around 1 million individuals from Friday Harbor waters. The population used to bloom massively every summer, hundreds of thousands harvested by scientists. Then in the 1990s and 2000s, the numbers crash. The population becomes relatively scarce. The cause remains unknown, not overcollection. Mid-1980s. Martin Chalfie at Columbia University works with transparent roundworm Caenorhabditis elegans, trying to track gene expression. He attends a lecture mentioning GFP. Inspiration hits. Insert the DNA sequence for GFP into worms and they'll glow wherever that gene expresses. 2008, Shimomura, Chalfie, and Roger Tsien share the Nobel Prize in Chemistry. The discovery revolutionizes cell biology. Researchers can now track proteins, gene expression, cellular processes in living organisms in real time, all from a transparent jellyfish that barely registers when you shine a light through it. Ships carrying ballast water from the Gulf of Mexico discharge into the Black Sea. The Catenophore arrives. 1982, first recorded. Common name sea walnut or warty comb jelly. Transparent lobed body, 8 rows of ciliated combs running vertically. They glow blue-green when disturbed. Body comprises 97% water. Maximum length, 7 to 12 centimeters. Diameter, 2.5 centimeters. It tolerates salinity from 2 to 38 practical salinity units. Temperature from 2 to 32 degrees Celsius, 36 to 90 degrees Fahrenheit, 1989. First massive bloom near northeastern Black Sea coast. Biomass hits 840 million tons. The entire basin becomes infested. The animal eats zooplankton, copepods, fish eggs, fish larvae. No natural predators in the Black Sea. Reproduction explodes. Egg production starts when animals reach 15 millimeters. Size determines output. 2 to 3,000 eggs per day at high food concentrations. The species undergoes reverse development, returns to previous life cycle stages, functional immortality. Transient anus appears only during defecation, no permanent connection between gut and rear of body. Waste accumulates. Part of gut balloons until it touches outer epidermis. Fusion creates temporary anal opening. After excretion, the anus vanishes. Adults defecate once an hour. Larvae every 10 minutes. Commercial fish populations collapse. Anchovy catches plummet. The ecosystem shifts from fish-dominated to gelatinous-dominated. The invasion spreads to Azov Sea, Caspian Sea via oil tanker ballast water, Baltic Sea, North Sea, Mediterranean Sea. By 2009, the species reaches the Ligurian, Tyrrhenian, Ionian Seas. 2010 genetics reveal source. Gulf of Mexico populations match Black Sea populations. Secondary dispersal occurs from Black Sea to Mediterranean. Venice Lagoon, Italy records large blooms. Artisanal fisheries report severe impacts. Interviews with fishers date blooms. Landing records analyzed. Catches decline. Composition changes. The ctenophore now listed among the 100 most dangerous invasive species by the International Union for Conservation of Nature. Its genome sequenced, 10 kilobases, smallest animal mitochondrial DNA known, lost 25 genes, including ATP6 and all transfer RNA genes. Relocated ATP6 gene to nuclear genome, acquired introns and mitochondrial targeting sequence. Original phylogenetic analysis suggested ctenophores as sister lineage to all other animals. Later studies debate. Position of Ctenophora still contested. It moves so slowly, researchers call it sea walnut. Evolutionary lineage lost features and magnified others. Solitary individuals act as motherships, asexually breed chains of clones. Colonies of hundreds linked together, chains several meters long. University of Oregon researchers, led by Kelly Sutherland, published findings in Science Advances May 15, 2024. The study examines salp migration, vertical migration from deep ocean to surface. It happens every night. Largest migration on planet by biomass. Researchers use special cameras for high contrast imaging against black backdrop. They encounter immense flurries during nighttime dives. Salps perform daily marathons using novel fluid mechanics. 2 swimming modes observed. Shorter colonies spin around an axis like spiraling football. Longer chains buckle and coil like corkscrew. Helical swimming, contracting muscle bands like human throat muscles. Pump water from one body side, squirt from other end, creates thrust, different from microorganisms that beat water with hair like projections or tail whips. Salps use genuine jet propulsion. Research aims to inspire robot design for deep sea traversal. Energy-efficient movement through water column. Salps are macroplankton, live far from shore, some solitary, some colonial. Bodies filled with water, transparent except for small visible organs. They filter feed, consume phytoplankton and small particles, play major role in carbon cycling. Their fecal pellets sink fast, transport carbon to deep ocean, climate relevance high. Predators include sea turtles, fish, and the amphipod Phronima, which hollows them out for housing. The chains coordinate movement. Each individual pulses in sequence, creates wave motion through entire colony. Propulsion efficiency increases with chain length. They avoid predators through transparency and coordinated escape responses. Population blooms can be massive. When conditions favor reproduction, salp numbers explode. They outcompete other zooplankton. Shift food web dynamics, then population crashes when food depletes. 24 families, 156 genera. Arose in Cretaceous period over 140 million years ago. Includes conger eels, moray eels, garden eels, all true eels of anguilliforms. Also tarpon, bonefish, spiny eels, pelican eels. Deep-sea species like Chima atrum, laterally compressed bodies, transparent jelly-like substance inside, thin muscle layer outside with visible myomeres, small body organs, simple tube gut. Combination creates extreme transparency when alive. Dorsal and anal fins confluent with caudal. No pelvic fins. No red blood cells until metamorphosis into glass eel stage. Fang-like teeth present until metamorphosis, then lost. Leptocephali grow much larger than most fish larvae, long larval periods, 3 months to over 1 year. European eel anguilla spawns in Sargasso Sea. Danish oceanographer Johannes Schmidt establishes this in 1920s. He surveys larvae, finds increasing size with distance from Sargasso. Breeding area approximately 1 million square kilometers. Larvae drift. American eels head to North America. European eels to Europe. Migration distance up to 9,000 kilometers. Migration duration disputed. Schmidt's data suggested 3 years based on 3 length classes present simultaneously. Passive drift estimates agree, 2 to 3 years. But otolith daily growth rings indicate 6 to 7 months. Larvae live primarily upper 100 meters at night, deeper during day. They feed on marine snow, tiny particles floating in ocean, particulate organic matter. Includes discarded appendicularian houses, zooplankton fecal pellets, protists, amorphous materials. DNA sequencing shows gut contents from wide range of microorganisms and food web species. Isotopic studies confirm low trophic position. Laboratory rearing attempts showed larvae extremely selective. Japanese eel Anguilla japonica reared from newly hatched preleptocephalus at 3.6 millimeters to maximum size, 59 millimeters at 260 days old. Researchers used shark egg yolk in artificial diets. Larvae would eventually eat specially formulated diets. Wild-caught leptocephaly intestines usually empty when collected. The transparency in fragile bodies means standard plankton nets don't catch them well. Good swimming ability helps them avoid capture. A video recording off Hawaii at night shows natural swimming. Rod-dominated visual system. Some families, like Cynophobranchidae, have telescopic eyes. Tubular eye with sphere-shaped lens. The larvae represent extreme adaptation to pelagic existence. Bodies filled with glucosaminoglycan compounds. Geiagi transforms into adult tissue during metamorphosis. Also stores energy. Maximum larval size varies. 5 to 10 centimeters common. Can reach 30 centimeters or more depending on species. After maximum size, they enter glass eel stage. Bodies become more rounded. Head thickens. Olfactory organ enlarges. Teeth lost. Then they migrate to recruitment habitats. Rivers, estuaries, coastal areas. European eel larvae not identified as such until 1896. Italian scientist Giovanni Grassi observed metamorphosis of what was classified as separate species Leptocephalus brevirostris into glass eel, revealed second life stage of European eel. Usually about 2.5 centimeters long, 1 inch, resembles shrimp with head, eyes, jaws, clawed arms, 4 eyes instead of 2, 2 pairs situated on each side of head, outer and inner eye in contact, outer eye surrounded by lozenge-shaped ball of eye facet lenses. Field of view perhaps 270 degrees. 1 pair watches ahead, other pair views surroundings through transparent walls of temporary home. Inspiration for xenomorphs in Alien film series. Director James Cameron's Aliens from 1986 shows alien queen with similar morphology. Broad crest to top head, tubular eyes, parasitoid behavior. Phronima females attack salps and other gelatinous organisms. Use mouth and claws to eat the animal. Hollow out gelatinous shell. Female enters barrel. Lays eggs inside. Propels barrel through water as larvae develop. Provides fresh food and water. The creature devours living tissue from tunicate. Leaves barrel-shaped house. Lives inside. Drives it through deep ocean searching for prey. Offspring grow and hatch along inside walls. Pram bug, Kinderwagen. Krebs in German, baby carriage crab. The transparency helps avoid predators in sunlit waters. It can contract pigmented cells called chromatophores, appear completely transparent. Prey mistake it for harmless gelatinous plankton, then claws strike. Carnivorous, eats small plankton, found at great depths usually by submarine crews. Eyes adapted to detect blue-green wavelengths, prominent in bioluminescent flashes of prey and predators. Most hyperide amphipods live in groups. Phronima, solitary, free-floating. Jelly barrel provides protection from predators, storage for food, space to raise young. Researchers at Smithsonian Institution study vision mechanisms using computer models. Karen Osborne, research zoologist at National Museum of Natural History, works on understanding how 4-eyed crustacean sees in Twilight Zone. Twilight Zone spans a light-flooded surface and perpetually dark abyss. 800 to 1,200 meters, typical Phronima depth range. The transparent body combined with gelatinous home makes detection nearly impossible. Perfect camouflage for perfect predator. More soluble than calcite. Live in temperatures minus 0.4 to plus 4 degrees Celsius, rarely up to 7. Vertical distribution by size. 0.2 to 0.4 millimeter individuals live 0 to 50 meters depth. Larger pteropods 0 to 150 meters. Innumerable quantities noted by Constantine John Phipps in 1774. Major component of polar zooplankton. Produce large mucus webs to filter feed. Eat web with captured prey. Reproduce web. Web large, spherical, difficult to see during day. Pteropod, Cleone limicina, feeds almost entirely on genus Limicina. Also Pedicleone duliiformis, but only juveniles with shells smaller than 1 millimeter. They fly through water, wing like parapodia, beat in complex 3D stroke pattern, resembles flying insect kinematics. High angle of attack 45 to 50 degrees generates lift. Wings beat 4 to 10 times per second. Uses clap and fling mechanism described by Torkel Weiss Fogg and small insects. Extreme forward back pitching during each half stroke. Hyper pitching. Ocean acidification threat level critical. Anthropogenic carbon dioxide dissolves in ocean, makes water more acidic. Calcium carbonate dissolves more easily. Since Industrial Revolution, 40% of emitted carbon dioxide absorbed by oceans. Aragonite saturation, state falling. When omega aragonite drops below 1 dissolution, thermodynamically favored. Researcher Nina Bednarsek from Southern California Coastal Water Research Project presents findings at Alaska Marine Science Symposium. Field observations show dissolving shells from Gulf of Alaska to Beaufort Sea. Western Gulf of Alaska encompasses spawning grounds. 70% of animals in Canadian Beaufort Sea fjords have corroded shells. Glacial meltwater exacerbates acidification. Laboratory studies warned for decades, now happening in wild. 2011 surveys off U.S. West Coast find 24 to 53% individuals with damaged shells. Shell dissolution tracks closely with regions of acidified water. Pteropods patch damaged shells, but at cost, physiologically compromised. Energy diverted to shell rebuilding. Triggers stress responses. Critical food web roll. Pink salmon to whales feed on them. Fish depend on pteropods. People depend on fish for subsistence. Protective outer coating periostracum. Researchers debate effectiveness. Some studies show periostracum protects when intact. Damage to coating results in dissolution when exposed to corrosive water. Specimens collected from Greenland Sea, June 2012, show dissolution localized where periostracum damaged, where coating intact, shell pristine. Micro-CT scans reveal repair mechanisms. Despite losing entire thickness of original shell in areas, Limacina helicina maintains integrity by thickening inner shell wall. One specimen from Fram Strait generated 4 times original thickness in repair material. Metabolic cost high. Overwinter calcification efforts focus on repair and maintenance. Food scarcity during winter compounds problem. Sea ice seasons doubly troublesome. Exposed aragonite dissolves while food scarce. Rate of internal repair calcification must keep pace with dissolution. Metabolic upregulation energetically demanding. Diminishing limit to repair capacity under further undersaturated conditions exists. Aragonite saturation, state 1.5 threshold proposed. Quantitative relationships between saturation state and transparency explored. Climate change accelerates process. More carbon dioxide means more acidic oceans means more shell dissolution. The canary in coal mine already dying. The ice fish with blood as white as snow and a see-through brain. 1928. Norwegian zoologist Johan Ruud examines fish specimens caught near South Georgia Island, Southern Ocean, Antarctica region. He cuts one open. The blood runs clear, cloudy white, not red. Initial assumption? Specimen degradation, poor preservation, but more specimens. Same result, every single one. Colorless blood, white hearts, translucent tissue. Ruud publishes findings in Nature. The scientific community reacts with shock. This contradicts fundamental vertebrate biology. Every vertebrate on Earth has hemoglobin. The protein that makes blood red carries oxygen through the body, required for survival, except these fish. Family Chanectidae, crocodile ice fish, white-blooded fish, at least 16 species currently recognized, 8 additional species proposed. February 2021, scientists discover breeding colony, Neopagitopsis iona species, 60 million active nests documented, Weddell Sea seafloor, Antarctica, 92 square miles of nesting area. Largest fish breeding colony ever found. The fish have no hemoglobin in blood, no red blood cells in circulation. Genetic analysis reveals both alpha and beta hemoglobin genes deleted. 15 of 16 species show complete beta subunit gene deletion, partial alpha subunit deletion. One species, Neopagitopsis iona, retains more complete beta subunit, but still non-functional. Cannot produce working hemoglobin protein. Red blood cells, usually absent. When present, extremely rare and defunct. Non-functional. Oxygen transported entirely via plasma, dissolved directly in blood fluid. Fish survive because metabolism runs extremely slow. Environmental oxygen solubility high in cold water. The colder the water, the more dissolved oxygen it holds. Physics and chemistry enable impossible biology. Oxygen carrying capacity of icefish blood, less than 10% compared to relatives with hemoglobin. Myoglobin also absent from skeletal muscles. The protein that stores oxygen in muscle tissue, gone. However, 10 species retain myoglobin in heart, ventricles specifically. Loss of myoglobin gene expression occurred at least 4 separate times across evolution. Compensations evolved. Larger blood vessels throughout body, capillaries expanded to double normal diameter, low viscosity blood because no cells blocking flow, very high flow rates possible at low pressure, blood volume 4 times greater than other fish, hearts enlarged, 5 times greater cardiac output compared to related species with red blood. Hearts lack coronary arteries entirely. Ventricle muscles extremely spongy, porous, absorb oxygen directly from blood they pump. No need for separate arterial blood supply to heart muscle. First, ice fish discovered in 1920s called white crocodile ice fish. See through body. Can see brain through skull. Big eyes, long snout full of teeth. Resembles crocodile head. Since then, 16 species identified. Fish grow to 30 to 44 centimeters, 1 to 1.5 feet long. Live 4 to 5 years typically. Mackerel ice fish, Champsocephalus gunari, found around Heard Islands, McDonald Islands, Kerguelen, South Georgia. Once most abundant species in shallow waters less than 400 meters. Population crashed 1970s and 80s, overfishing documented. Commercial harvest depleted stocks, recovery slow, fish form schools, migrate vertically every 24 hours, spend day near seafloor depths, feed on plankton and small fish, including juvenile ice fish, cannibalistic when food scarce, move to midwater at night, important food source for seabirds, seals, large fish, Antarctic toothfish. Disosticus mawsoni preys heavily on ice fish. Evolutionary history debated. 2 competing hypotheses. First suggests ice fish only 6 million years old. Appeared after Southern Ocean cooled significantly. Second proposes 15 to 20 million years old. Formation of Antarctic polar frontal zone and Antarctic circumpolar current marks beginning of Antarctic fish evolution. Current moves clockwise northeast direction. Width up to 10,000 kilometers, 6,200 miles. During mid-tertiary period, species crash opened empty ecological niches. Hemoglobin loss mutation normally lethal, but lack of competition allowed mutants to survive. Colonize empty habitats. Evolve compensations for mutations. Later periodic fjord openings created habitats colonized by few individuals. Genetic bottlenecks. Loss of myoglobin occurred during this time. Initially, scientists believed hemoglobin loss was cold adaptation. Decreased blood viscosity in cold temperatures. But further analysis refuted this. Hemoglobin loss, not adaptive, but genetic accident. Deletion occurred approximately 8.5 million years ago. Specific mutation deleted beta-globin gene cluster. Additional mutations accumulated disabling alpha-globin genes. Such mutations lethal in any other environment. But oxygen-rich, near freezing Antarctic waters allowed survival. Prime example of extreme environment driving extreme adaptation through natural selection. Bill Detrick from Northeastern University sequences ice fish genome, confirms virtually all hemoglobin genes absent. Chicken versus egg, question remains. Did fish lose red blood cells first, then couldn't express hemoglobin? Or lose hemoglobin genes, then red cells withered? Research continues. Southern Ocean essentially saturated in dissolved oxygen. Stormy conditions mix water column constantly. Oxygen levels stay high. But new threat emerges. Climate change. Southern Ocean getting warmer, more acidic, less nutritious. Ice fish more sensitive to temperature changes than red-blooded fish. Cannot tolerate heat. Warming waters threaten survival. Species evolve for sub-freezing temperatures, face a habitat unsuitable to physiology. Clear blood, transparent tissue, white hearts. Ghostly appearance in icy depths. Ultimate transparency, taken to genetic level, blood itself invisible. Blue whale weighs 170 tons. Siphonophore weighs grams. Body diameter no bigger than broomstick. 2.5 centimeters. 1 inch thick maximum. This isn't one animal. Colonial organism. Superorganism. Intimately connected colony of tiny creatures called zooids, each zooid genetically identical, all developed from single fertilized egg, but morphologically specialized for specific functions. Some zooids form tentacles for catching prey, others become swimming bells for propulsion, others transform into feeding organs with mouths, reproductive structures for spawning, defense mechanisms armed with stinging cells. Zooids cannot survive independently, rely completely on symbiotic integration. Complete specimen needs all parts functioning together. Zooids arrange themselves in long stem structure, chains stretching through deep water. Monterey Bay. Aquarium Research Institute observed living Priadubia in 1987 for first time. Systematic study of water column off Monterey Bay, California. Natural habitat, direct observation. Prior to this discovery, remains dredged up in fishing nets resembled shapeless blob of gelatin. prevented proper identification as unique creature until 19th century. The animal swims actively, not passive drifter like jellyfish. Attracts prey using bright blue bioluminescent light, intrinsic biological luminescence. When finds region abundant with food, holds position steady, deploys curtain of specialized tentacles covered densely with nematocysts, stinging cells packed with toxin. Produce powerful sting, paralyzes or kills prey instantly upon contact. Diet includes gelatinous sea life like salps and small jellies. Small crustaceans including copepods. Possibly small fish and fish larvae. No known predators documented by researchers. Lives primarily in mesopelagic to bathypelagic zones. 700 to 1,000 meters below surface typically. 2,300 to 3,300 feet depth. Core habitat between 75 and 127 meters in some documented regions. Found off coasts worldwide in all major oceans. Iceland in North Atlantic. Chile in South Pacific. Japan, Australia. Circumglobal distribution confirmed. Different specialized zooid types identified by microscopy. Some have long tentacle for catching and immobilizing food items, distributing digested nutrients throughout entire colony via connected tissue. Others, called palpons or dactylozoids, contain excretory system structures, may assist in colony defense. Precise function in Praia dubia remains poorly understood. Transparent leaf-shaped bracts, also called hydrophilia, generally classified as another zooid type variant, covers and protects other more vulnerable zooids, forces rapid contraction in times of danger. Pneumatophores contain gas mixture, provide flotation and buoyancy control. Nectophores are structures that expel water forcefully, propel entire colony through water column, function as biological motors or jet engines. Gastrozooids serve as mouth and stomach of colony, feeding polyps with digestive capability. Dactylozoids function as tentacles equipped for dual defense and active hunting, armed densely with stinging cells containing toxins. Hydrostatic skeleton held together entirely by ambient water pressure, requires pressure above 46 megapascals, 460 bar minimum. Considerable pressure only exists at depth. When brought rapidly to surface, specimens burst catastrophically, but yield only amorphous jelly-like mass unrecognizable. True shape and delicate structure remained completely unknown to science for over a century, until living specimens finally observed at proper depth underwater using submersibles. Body color varies significantly by depth habitat. Deeper dwelling individuals appear distinctly redder in coloration. Shallow water individuals, more translucent, white, or colorless. April 2020. Schmidt Ocean Institute expedition discovers remarkable specimen, 150 feet long documented, longest siphonophore ever recorded by science, spiraling specimen from related Apolemia genus. Some marine biologists argue Praia dubia, actually longest animal in world altogether. Others counter that colonial animals shouldn't directly compare with singular organisms like whales. Scientific debate continues unresolved. Siphonophores represent important predators throughout ocean ecosystems worldwide. Drift suspended in open water columns, spread elaborate specialized tentacles wide, trap unsuspecting prey swimming into range. Slightest physical touch triggers tentacle rapid recoil reflex. Snares meal within fraction of second. 175 described species total according to World Register of Marine Species database. Most exhibit similar fundamental body plan design. Gas-filled float structure for buoyancy control, swimming bells for active propulsion through water. Central stem bearing specialized parts, feeding structures, defense mechanisms, reproduction organs. They reproduce asexually through budding process primarily. Single bud called probud initiates entire colony growth, undergoes repeated fission divisions, each zooid produced genetically identical initially. Random mutations can alter zooid functions over time, increases functional diversity within single colony. Siphonophora is unique among animals because probud initiates production of diverse specialized zooids with dramatically different specific functions. Function and precise organization varies considerably among different species. Majority arranged bilaterally with distinct dorsal and ventral sides to stem. Some species exhibit rare red bioluminescence capability. Irena, Serena species, documented producing red light. Only second life form ever found producing red bioluminescence. First being scaleless dragonfish chirostomias. Most siphonophore species produce standard blue-green bioluminescent light. Matches wavelengths, most prevalent in deep ocean environment. Used to attract prey toward tentacles, possibly communicate between distant zooids. Fluorescent tentacles twitch and flash rhythmically. Lure curious victims directly into lethal stinging range. When longer siphonophores manage staying intact reach truly remarkable lengths. Several members of Praia and Apolemia genera rank among longest animals world. Giant siphonophore, Praia dubia. Woolly siphonophore variants. various spiraling specimens documented, completely transparent in natural state, absolutely lethal to small prey, longer than any single multicellular animal except certain marine worms. Invisible floating death traps stretched across miles of deep ocean, technically classified as plankton by scientific definition, any creature passively drifting with ocean current, regardless of size, despite enormous total length, meets classification criteria perfectly. Glass squids film for first time in 2025 after ice breaks from warming. Bear-life fish navigating through jellyfish blooms that increase with ocean changes. Crystal jellies disappearing from waters where they once numbered hundreds of thousands. Sea walnuts invading new oceans via ships moving through routes opened by melting ice. Salps blooming in massive numbers when conditions shift. Eel larvae drifting through oceans where currents alter with temperature. Phronima hunting in twilight zones where light penetration changes. Sea butterflies dissolving in waters that turned acidic from human carbon emissions. Transparency evolved as ultimate camouflage. Can't eat what you can't see, can't be eaten if you're invisible. Millions of years of selection pressure. Bodies that bend light, internal organs miniaturized. Blood without color, structures filled with clear gel. Every species found its own path to vanishing. Squids with jellyflesh, fish with fluid-filled heads, jellies with tissue so thin light passes through, amphipods like living glass, eel larvae that look like ribbons. It worked. Transparency became dominant strategy in open ocean. But the game changed in 1 century. The water chemistry shifts, the temperature rises, the currents alter, the ice melts. Species that spent eons invisible suddenly can't cope with rapid change. Their shells dissolve, their prey disappears, their habitats shrink. The transparency that protected them can't shield them from chemistry. Another transparent form, unknown species, body structure unlike any cataloged. Researchers tag coordinates, sample water chemistry at depth, temperature 3 degrees Celsius, pressure 300 atmospheres. The organism drifts, almost invisible. Possible new genus, tissue samples collected, DNA sequencing pending. The ocean holds more transparent species than we've found. Conservative estimates suggest thousands remain undiscovered. Deep zones unexplored, midwater regions barely sampled. Every expedition reveals creatures science didn't know existed, each one adapted to invisibility in a unique way, each one vulnerable to changes happening faster than evolution can respond. The glass sculptures still drift through the depths, still hunting, still reproducing, still transparent. But the water around them changes, and transparency can't make you invisible to chemistry. Species we've known existed for over a century, and we're only now seeing them in their habitat. The ocean keeps its secrets well, but secrets don't protect against dissolved carbon dioxide, against warming currents, against acidifying water. The transparent creatures evolved in an ocean that stayed relatively stable for millions of years. They're living through changes happening in decades. That's the equation. Millions of years of adaptation versus decades of disruption. Watch the footage from 2025. The colossal squid juvenile drifting, the glacial glass squid spreading its arms, the barrelay rotating its eyes, the crystal jelly pulsing, the sea walnut blooming, the salt chain spiraling, the eel larva ribbon swimming, the amphipod in its barrel, the pteropod with dissolving shell, everyone transparent. Everyone struggling with changes they can't adapt to fast enough. The ocean's invisible kingdom is becoming visible, not because we developed better cameras, but because the chemistry is making their shells dissolve and their populations crash and their habitats vanish. You're seeing them now. The question is whether your children will.